In this video, I will be going over brain imaging techniques with a focus on describing, evaluating, contrasting, and discussing MRIs and fMRIs. I will also go over a research for each technique. So, from the time phrenology was a practice, scientists, doctors, and even philosophers have tried understanding the structure, function, processes, and influence of the brain on behavior and cognition. And for centuries, these researchers have been dependent and limited to postmortem autopsies. Brain imaging techniques, upon their introduction, really revolutionized and broadened the scope of psychological research. Today, brain imaging techniques can be used to understand structure and function in living beings through non invasive procedures, which wasn't possible in the past. Before we go into details about the two techniques that I mentioned earlier, I'd like to go over what resolution of brain images are. So there are two types of resolution: spatial resolution and temporal resolution. Spatial resolution is the ability of a scanner to capture differences between nearby locations or parts. So, with higher spatial resolutions, the image is able to capture more details. And on the other hand, temporal resolution is the shortest time period in which a brain scan can register changes in the brain activity. This is relevant to techniques that record brain activity, such as CT scans, fMRIs, and PET. So, the magnetic resonance imaging is the first technique that we're going to be looking at in this video. Okay. The brain is composed of 70% water in its tissues, and the magnetic resonance imaging, or MRI, takes this into account for its functioning. People are placed into a donut shaped scanner that emits a magnetic field, a really strong magnetic field. Okay, and it kind of looks like the image on the right side. This magnetic field then causes the hydrogen protons in the brain to realign. And when these protons realign, they emit energy, which is what is detected by the scanner. An MRI scan can last for about 15 minutes to as long as 90 minutes, depending on the number of images being taken and the region of the brain that the researchers are trying to focus on. Okay. The scanner produces a composite image of brain structures. What this means is that it takes multiple images that are put together and are analyzed using pixel counting and voxel-based morphometry. Okay, so pixel counting really measures the area of the brain structure. Okay, so if we're looking at the hippocampus, so we're trying to look at the area of the hippocampus. And voxel-based morphometry, which is also VBM, measures the density of gray matter. Okay, the final image that an MRI produces is three-dimensional. Okay, so it's a 3D image that can be viewed from multiple angles. Okay, and also in slices. MRI scans are usually used to investigate brain damage, comparing the brains of different individuals, such as the London taxi drivers, in comparison to healthy controls. Okay, and also to track brain development through lifespan or in relation to a specific task, such as in Draginsky's research. They use the MRI to track the development of the brain in relation to learning juggling as a new skill. Okay. As for the strengths and limitations of MRI scans, it has high spatial resolution, so it is able to capture details and differences of nearby regions. Okay, so it is this feature of the MRI that allows us to see whether there is a redistribution of matter in the brain in a specific area, such as the hippocampus. Okay, and even within the hippocampus, we're able to see whether there is a change in the anterior and the posterior hippocampus parts. Okay, so having high spatial resolution allows researchers to then see these details and kind of analyze these details. Okay, another Strength of the MRI is that it is a non-invasive technique and it involves no harmful radiation, unlike other brain imaging techniques such as the PET. On the other hand, researches that utilize MRI scans tend to be correlational in nature unless it is conducted over a period of time. Okay. Let's say we conduct, if we take, for example, Maguire's research, there was only one MRI scan that was conducted on the participants. Okay, so that then indicates the person's brain structure at that given point of time. Okay, so we don't know what their brain structure was like prior becoming taxi drivers. We don't have much of a comparison there. Okay, so because of that, a lot of the results tend to be correlational. Okay, if there is a longitudinal research that is conducted, the potential to establish cause-effect relationships does increase. But again, that could also involve a lot of extraneous variables influencing the relationship. Okay. And another drawback of the MRI scan is that individuals who have any type of metal in their body or are claustrophobic, they cannot undergo the procedure. Okay. It is also not ideal for young children as the scan requires participants to remain really still in the scanner, okay, for a long duration. So another limitation is that it is expensive, which usually influences the number of participants recruited for the research. And when there is lesser participants, generalizability of the findings is affected. Okay, and also the results that are finally arrived at can be attributed to chance and not actually, the IV having an effect on the DV. So, a research that utilizes MRI scans is Maguire et al.'s London taxi driver research. This is a great research to use, okay, for brain imaging techniques. You can use it for localization and even neuroplasticity. So, it is always good for you to remember such researches that have overlaps, okay, because at the end of the day, it then helps you minimize how many researches you need to remember in total. So, the aim of Maguire's research was to investigate whether there were differences in the brain structure of London taxi drivers when compared to the average brain. MRI scans of 16 right handed male taxi drivers who had their license for at least 1.5 years were compared to those of 50 healthy right handed male participants. Okay, and this was collected randomly from an MRI database, and the MRI scans of these participants basically were of people who had no taxi driving experience. So the pixel counting in this research indicated that there was no significant difference in the overall area of the hippocampus, okay? However, there were regional differences. So basically within the hippocampus, they saw differences. The posterior hippocampus was significantly larger in the taxi drivers, and the anterior hippocampus was larger in the control group, okay? Basically the participants who were non-taxi drivers. Furthermore, the voxel-based morphometry, basically the VBM, revealed that the density of gray matter in the taxi driver's hippocampus was positively correlated with the amount of time that they spent taxi driving. So, what that basically means is that a taxi driver who has driven for 8 years will have a denser hippocampus than one that has driven for only 2 years. Okay, so the more you've driven, the more dense your hippocampus is. So, as you can see in this research, the MRIs have allowed the researchers to investigate and compare the structure of the brain in living participants. It also provides evidence for redistribution of brain matter in relation to a specific experience or skill, which is here taxi driving. Okay, and it also helps developing the understanding that the posterior hippocampus is involved in using previously learned spatial information, whereas the anterior hippocampus is responsible for maybe learning new spatial information. As for the evaluation, one of the major drawbacks of Maguire's research is that it is cross-sectional and not longitudinal. The MRI scans are indicative of the brain structure of the participants at that given point of time. For this reason, the relationship between the taxi driving and the difference in hippocampus is only correlational. Okay, it is possible that the redistribution may have occurred in response to other stimuli and other experiences that the taxi drivers have had. There's also potential for bidirectional ambiguity, okay, wherein individuals who have a greater or larger posterior hippocampus have better spatial memory and related skills, okay, and for that reason chose to become taxi drivers. So we don't know the direction because the relationship is only correlational. However, this is less of a concern in this particular research since there was a correlation established between the number of years of taxi driving experience and the increased density of gray matter in the hippocampus. So because there was kind of a relationship between the number of years or the amount of time spent taxi driving with gray matter density increasing, okay, it does show that there is kind of a direction. So this then suggests that there is a causality or potential causality between taxi driving and changes in the hippocampus. Moving on to the functional magnetic resonance imaging, or fMRI. The fMRI is an upgrade of the MRI technology and follows similar principles and technology. Participants are placed in a donut-shaped device and asked to do a task such as solving a puzzle, visualizing a memory, or watching a video. This advancement of the MRI takes into account that when a region in the brain is active, there is an increased supply of oxygenated blood. Okay? Oxygenated blood is diamagnetic and deoxygenated blood is paramagnetic. We don't really need to know what that is, but basically there's different magnetic properties or of oxygenated and deoxygenated blood, and the fMRI is able to detect these changes and produce an understanding of the brain activity in relation to a specific task that they're doing or stimulus that they're exposed to. This mechanism that the fMRI uses is referred to as blood oxygen level dependent, and the fMRI is different in that it is able to capture activity in brain regions in color coded images. Okay, and the changes in brain activity can be captured per second. Some points of strengths and limitations for the fMRI. The main strength of the fMRI is that it has both high spatial and temporal resolution. So researchers are able to record activity every second, as I mentioned before, and also in specific details. Okay, so again, if we wanted to focus on the hippocampus and we wanted to look at the activity in the posterior hippocampus, the fMRI would be able to do that. There is potential to establish cause-effect relationships using this technique. However, this is often an assumption, okay, that the changes that we are observing in the images is due to the stimulus that the researchers are introducing to the participants. And another point is that the establishment of cause-effect relationships in the use of fMRIs are affected by the fact that the scan can be easily influenced by artifacts, okay, and also some extraneous variables such as, noise, anxiety, being in an artificial environment, and so on. This then results in the ecological validity of the research being compromised when using fMRIs. One research that you can use to support the use of fMRIs is Fisher, Aron, and Brown, 2005. These researchers used the fMRI to investigate the neurobiological correlates of romantic love. This research can also be used for neurotransmission, specifically to support the influence of dopamine on love. The participants were 17 young adults who were screened for the duration and intensity they were in love with their partner for. They were then placed in the fMRI scan and asked to view images. First, they viewed an image of their loved one for 30 seconds, followed by a distraction task for 40 seconds, followed by another image of a neutral acquaintance, and the final task was a 20-second filler task. It was found that when the participants were exposed to the image of their loved ones, the right ventral tegmental area, the VTA, and caudate nucleus were activated. Okay, and this was in comparison to when they saw the image of the neutral acquaintance. And the neutral acquaintance image kind of serves as a baseline. Okay, so the VTA is a dopamine rich area of the brain and is a part of the reward system responsible for pleasure, arousal, motivation, and the caudate nucleus is responsible for detecting and expecting rewards. This then basically suggests that dopamine plays a role in the motivation and expected rewards of pursuing romantic love. So, the high temporal and spatial resolution of the fMRI allowed the researchers to observe changes per second in specific brain areas over the span of the few seconds that they had the procedure. Okay. Another strength of this research is that it has filler tasks in between. Between when they saw the image of their loved one and then the mutual acquaintance. Okay, so considering the high temporal resolution of an fMRI, if there weren't any filler tasks between the exposure to the two different images, there could have been carryover effects given the repeated measures design of the experiment. Okay, so maybe if there was no filler task and they just showed the loved one and then the neutral acquaintance, they wouldn't be able to distinguish between the activation in response to the loved one and the activation in response to the neutral acquaintance. By using this repeated measures design, the researcher also controls for individual differences in brain activity, as it is measuring activity of the same person when exposed to both a romantic partner and an acquaintance. The drawback, on the other hand, is that the changes could be in response to other extraneous variables or even artifacts, such as excitement or of being a part of the experiment, anxiety, and so on, which would have easily influenced the brain activity of the participants. So that's kind of about it with regards to the topic itself. But when it comes to command terms, one of the command terms that you might have for your LAQ is discuss. And when it's a discuss question or a discuss command term, it is not enough to just evaluate the research. Okay, you have to discuss the use of the technique. Okay, so it's kind of a good idea to look at the broader topic or also the body of research in that you are using in your essay. Okay, so here are some points of evaluation or points of discussion when it comes to brain imaging techniques. The first point is that the MRI and fMRI are indirect measures of brain structure and brain activity. If you look at EEGs, they use electrodes to record brain activity, which is in line with neuronal communication being electrical signals. Okay, MRI and fMRI use alignment and emission of energy from hydrogen atoms and molecules of oxygenated blood. So there is an assumption that these are related to the structure and activity, and this limits the degree to which we can confirm the relationship between the variables and also generalize the results. This is kind of an area of uncertainty when it comes to brain imaging. Okay, because these are indirect measures, there is a certain degree of assumption that yes, these are indicative of the structure and the function or the activity of the brain. As mentioned in the previous slides, fMRIs are influenced by artifacts and the artificial environment. Even with MRI scans, there are limitations to the image produced, even though it is a structural image of the brain, and that won't really change. Even the slightest of movements during the scan may distort the image produced. Okay, and this could affect the reliability of what a researcher may observe. there is subjectivity involved. Okay, so while the images do produce color-coded images, it has been up to researchers to interpret the meaning of these colors. Furthermore, the colors in these scans exaggerate the effects of the brain. They can be kind of misleading in that they suggest that specific regions are clearly defined when in reality, activity in the brain is not as localized as we make it out to be. Okay, so the brain is also active for several other reasons in addition to the stimulus that they're being exposed to. So it isn't necessary that the changes are in response to the stimuli. Okay, so all brain imaging techniques are fairly expensive, which results in small sample sizes across the board. Usually, most researchers that you come across will have low samples. So if you look at Maguire's research, there's only 16 participants, and the other participants, the other 50, were from a database, okay? And for Fisher's research, there are 17 participants. And this obviously affects the generalizability of the findings, and the lower the number of participants, it also kind of brings in the possibility that there are other factors that are influencing the relationship, okay, in addition to the IV and DV. And finally, the techniques we utilize in this day and age are far more ethical, okay, so on the positive side, they are far more ethical and they're also more sophisticated. So it helps us conduct research in a non-invasive manner, unlike the autopsies that were previously used. So these are a few points of discussion. Again, these are more general than in an evaluation of specific techniques. Okay, these would kind of apply to multiple techniques that are used in psychology. So if you are able to include some of these points of evaluation in your discussion or discuss command term question, that would be great, because it shows that you are able to look at the topic or the question more holistically and not just in relation to specific techniques being evaluated. The other possible command term is contrast, okay? A contrast command term is likely for this topic, wherein you would have to evaluate the differences between two techniques of your choice, okay? And again, I've chosen MRI and fMRI. So for a contrast question, your 6 points of critical thinking is awarded based solely on your points of contrast, okay? Evaluating each technique in isolation will fetch you lower band marks and you will lose points for focus as well. The first difference between MRI and fMRI is that MRIs take images of brain structure, okay, whereas the fMRI is able to image brain activity as well. So an MRI would be limited to investigating brain damage, comparing different brain structures, like in Maguire's research, okay, and tracking brain development either through lifespan or following the introduction of a specific stimulus, such as juggling in Draginsky's research. Localization can be inferred, but an fMRI would be better in terms of the options between the two. It would be better to observe localized brain activity. Okay. So when it comes to the mechanism of the two techniques, the MRI utilizes magnetic fields to realign the hydrogen atoms present in the brain, whereas the fMRI takes advantage of the fact that when a brain area is active, there is increased flow of oxygenated blood. This blood oxygen level dependent mechanism is able to detect the changes between oxygenated blood, which is diamagnetic, and deoxygenated blood, which is paramagnetic. Okay, so when there's a switch between these two, the machine is able to detect that. The image produced by an MRI is a static image. Okay, it is black and white and only indicates structure. It has high spatial resolution, but no temporal resolution. Okay, for these reasons, most researches that use MRI scans are correlational in nature. okay, as the scan is indicative of the brain structure at that given point of time. Again, if this technique is used over a time period, there is more potential to establish cause-effect relationships. In contrast, the fMRI is a dynamic image that is color-coded and records brain activity per second. Okay, it has both high spatial resolution and temporal resolution, so it is able to effectively image both structure and activity. So moving on, the next point is with regards to the influence of artifacts and extraneous variables. So the MRI is not affected by the presence of sound, participant excitement, and other artifacts in the environment. Okay, the only factor that would affect an MRI is if the participant was moving in the scan or during the scan. Okay, however, if investigating, let's say, neuroplasticity, it is possible that there are other extraneous variables outside the lab environment that would influence the relationship between the independent variable and the dependent variable. The relationship that is established, again, is only correlational. As for the fMRI, it is easily influenced by artifacts that are present in the lab environment, okay? And this is due to the high temporal resolution. So if there is a lot of sound, which is the case with the machine, okay, participant anxiety, distraction, all of these factors or all of these artifacts would influence the brain activity that is being recorded, okay? So regardless, the fact that it has the ability to record activity per second, the researchers are able to record changes in activity in response to specific stimuli, okay? And with multiple trials, there is more potential to develop a cause-effect relationship. So the final point is with regards to the artificial environment that both research using MRI and fMRI are conducted in. Okay, all of them are based in labs. So while the static MRI images do not raise concerns of ecological validity, because nothing in the lab environment is really going to influence the structure of the brain in that moment, right? So the use of fMRIs do in fact compromise on the ecological validity, as whatever responses the participants are exhibiting and the consequent brain activity that is being recorded is all observed in an artificial environment. So these are the main points of contrast between the MRI and fMRI. For a contrast question, again, you need to explicitly evaluate the differences between the two techniques in order to achieve higher band marks, okay? So that's it for this video. It has basically all that you need to know for brain imaging techniques. If you found this video helpful, do leave a thumbs up and subscribe to my channel for more IB psychology content.